Guest list heavy, big names abound. Academic and professional leaders, knowledge profound. Gary May, Robert Langer, Golden Sound. Podcast lit, wisdom's playground. Conversations rich, interviews bright. From MDs to artists, sharing insight. Chemistry vibe, conversations ignite. Steam dreams growing, future so bright. Chancellor May, Sidney Colley in the fold. M.K. Brown, Sheena Colley, stories told. Mike Zott, Robert Langer, wisdom gold. McCall Washington, young minds bold. Dr. Thomas N.G., Todd Golub, ignite. Abinaya Marcella, vision bright. Lesel Vincent, Sonola's insight. Michael Evans, cough under spotlight. Sarah Scrabalak, Nigella speak clear. Jake Desmond's vision future near. Mr. Bourne Kuna, Fortingo here. Darissa Ferguson, voices sincere. Guest list heavy, big names abound. Academic and professional leaders, knowledge profound. Gary May, Robert Langer, Golden Sound. Podcast lit, wisdom's playground. Conversations rich, interviews bright. From MDs to artists, sharing insight. Chemistry vibe, conversations ignite. Steam dreams growing, future so bright. L.C. Campo, Emery Brown's precision. Lynn Rashay Johnson brings thoughtful vision. Keshav Gavin transclinical mission. Dick Mann and Johansson research ambition. Louis Junheim, Trevor Johnson's art. And Pinder speaking from the heart. David Joshua Ferguson plays his part. Chadwin Hanna, Vincent Miranda start. Greg Zuckerman writing from the Wall Street beat. Cordell Hardy's wisdom research elite. Kyle Woom, Forbes list ambition complete. Gabriel Galarza's insights repeat. Guest list heavy, big names abound. Academic and professional leaders, knowledge profound. Gary May, Robert Langer, Golden Sound. Podcast lit, wisdom's playground. Conversations rich, interviews bright. From MDs to artists, sharing insight. Chemistry vibe, conversations ignite. Steam dreams growing, future so bright. Dev Mandavia, corporate strategy bold. Jenai Burrows, medical stories unfold. Doreen Duncan's vision, Island City mold. Paul Boyer's lectures, stories retold. Omar Sharif Ali, software meta. Economic talks, making minds better. Pranav Dorbala, med school go-getter. Ging Rahim Bia, engineering trendsetter. Savni Kulkarni, Abbott structural heart. Farm D candidate, giving advice to start. Podcast guest list, masterful art. New Chemist podcast, playing art part. Guest list heavy, names renowned. Knowledge deep, esteem, vibes profound. Podcast flowing, innovation sound. Future so bright, keep dreams unbound.
Welcome to the New Chemist podcast, one of the best chemistry podcasts globally. We are glad you are listening. We encourage you to subscribe and continue to listen.
Welcome to the New Chemist podcast. We are glad you are listening. We are one of the best chemistry podcasts globally. We encourage you to subscribe and continue to listen.
Welcome to the New Chemist podcast.
We are so glad you are listening. Feel free to subscribe on Spotify and tell your friends and colleagues about the podcast.
Our Deepest Fear by Marianne Williamson. Our deepest fear is not that we are inadequate. Our deepest fear is that we are powerful beyond measure. It is our light, not our darkness, that most frightens us. We ask ourselves, who am I to be brilliant, gorgeous, talented, fabulous? Actually, who are you not to be? You are a child of God. Your playing small does not serve the world. There's nothing enlightening about shrinking so that other people won't feel insecure around you. We are all meant to shine as children do. We were born to make manifest the glory of God that is within us. It's not just in some of us, it's in everyone. And as we let our own light shine, we unconsciously give other people permission to do the same. As we're liberated from our own fear, our presence automatically liberates others. Our Deepest Fear by Marianne Williamson. You are very important, especially to us here at the New Chemist podcasting group. You listening in is significant.
Vous êtes très important, surtout pour nous ici au New Chemist Podcasting Group. Votre écoute est significative.
Usted es muy importante, especialmente para nosotros aquí en The New Chemist Podcasting Group. Usted escuchando, es significativo.
Você é muito importante. Especialmente para nós do The New Chemist Podcasting Group. Você ouvindo, é significativo.
Είστε πολύ σημαντικοί, ειδικά για εμάς εδώ στο The New Chemist Podcasting Group. Το να ακούς είναι σημαντικό.
Sie sind sehr wichtig, besonders für uns hier bei The New Chemist Podcasting Group. Es ist wichtig, dass du zuhörst.
Je bent erg belangrijk, vooral voor ons hier bij The New Chemist Podcasting Group. Dat je meeluistert, is veelbetekenend.
You are very important, especially to us here at the New Chemist Podcasting Group. You listening in is significant. Welcome to the New Chemist Podcast.
Bienvenidos al podcast del nuevo químico.
Καλώς ήρθατε στο podcast του New Chemist. Welkom bij de podcast van The New Chemist.
Bienvenue sur le podcast du nouveau chimiste.
Bem-vindo ao podcast do novo químico.
Welcome to the New Chemist Podcast. Work hard. Be value-driven. You can do it. You can grow and learn it. You can be the difference you and your community needs. Don't give up. We are here, rooting and cheering for you. Don't give up.
Travaillez dur. Soyez axé sur la valeur. Tu peux le faire. Vous pouvez grandir et l'apprendre. Vous pouvez être la différence dont vous et votre communauté avez besoin. N'abandonnez pas. Nous sommes ici pour vous encourager et vous encourager. N'abandonnez pas.
Trabalhar duro. Seja orientado por valores. Você consegue. Você pode crescer e aprender. Você pode ser a diferença que você e sua comunidade precisam. Não desista. Estamos aqui torcendo e torcendo por você. Não desista.
Δούλεψε σκληρά. Να οδηγείτε στην αξία. Μπορείς να το κάνεις. Μπορείτε να μεγαλώσετε και να το μάθετε. Μπορείτε να είστε η διαφορά που χρειάζεστε εσείς και η κοινότητά σας. Μην τα παρατάς. Είμαστε εδώ για να σας ζητοκραυγάσουμε. Μην τα παρατάς.
Trabaja duro. Sea impulsado por el valor. Puedes hacerlo. Puedes crecer y aprenderlo. Usted puede ser la diferencia que usted y su comunidad necesitan. No te rindas, estamos aquí animándote y animándote. No te rindas.
Werk hard. Wees waardegedreven. Je kunt het. Je kunt groeien en leren. U kunt het verschil zijn dat u en uw gemeenschap nodig hebben. Geef niet op. We zijn hier om voor je te roten en te juichen. Geef niet op.
Work hard. Be value driven. You can do it. You can grow and learn it. You can be the difference you and your community needs. Don't give up. We are here rooting and cheering for you. Don't give up. Thanks for listening. We're glad you were able to tune into this podcast. Once again, this is The New Chemist, where we discuss chemistry, which simply put is the science of change, as well as the other sciences, careers, community, research, and COVID-19. Thanks again for listening. Note, the views on this podcast represent those of my guests and I.
In the world of pharmacy, we lay the claim. Knowledge and compassion is the golden flame. Science and practice, side by side they groove. In a student's journey, we find the move.
Purpose of these episodes not at all for advice or medical suggestions. These are aimed to provide support to peer pharmacists in training in educational and intellectually stimulating ways. Again, these are not at all for medical advice or medical suggestions. Please see your local board and state-certified health professional. The views of this podcast represent those of my guests and I.
Welcome to a deep dive. You ready to unlock some secrets of molecules? Really complex ones.
Sounds exciting.
We've got some awesome sources this time.
Oh, yeah.
We're going to be working with Dorothy Crowfoot Hodgkin. You familiar with her? Nobel lecture from 1964.
Okay, yeah, I've heard of her. That's going way back.
It is the early days.
Yeah.
Then we'll jump to 2009. Venki Ramakrishnan.
Oh, wow. Okay.
Excerpts from a lecture and some slides. He tackled the ribosome.
So.
That's a big one.
Huge. To round things out, we've got a 1965 lecture by Robert Burns Woodward.
The master.
Oh, yeah. Chemical synthesis. It's amazing to me how these pioneers, each working in their own time, you know,
right?
They help build this understanding of molecular structure.
And just pushing the boundaries of what was possible right at that time.
Absolutely. We're going to see how science evolves and builds upon itself. So let's start with Hodgkin. Her journey began with a simple spark of fascination.
Okay.
A children's book by W.H. Bragg.
Ignited her passion for visualizing molecules.
Wow. I love that. That's amazing.
Talk about finding your calling in unexpected places.
Really does. Speaks to that power of, clear, engaging science writing.
Yeah.
Even for these really complex topics. I mean, who knows how many, future scientists were inspired by that book.
It's true. And it led her to some incredible discoveries.
Yeah.
She started with simpler molecules, but eventually found herself at Cambridge working with J.D. Bernal.
Oh, wow.
His lab was like a hub. All sorts of research, digestive enzymes, pepsin.
Oh, wow.
Speculating on how muscles contract.
Oh, wow. That's amazing.
All sorts of things.
That's a fascinating lab. Bringing together people from all these different areas to really tackle these big questions. Must have been an incredible environment.
Sounds like a scientific playground.
Yeah.
Right? And it was there that Hodgkin truly mastered X-ray crystallography.
Okay.
So for anyone who's unfamiliar with this technique, imagine shining a super powerful light through a crystal where the light scatters. Yeah. And by analyzing that pattern, you can figure out the arrangement of atoms within the molecule.
It's like trying to figure out the shape of something just by looking at its shadow.
Yes.
But obviously much more complex.
Much more complex.
Yeah.
To make sense of these patterns, you need to understand electron density.
Right.
So this is essentially a map. It shows the distribution of electrons within the molecule.
Yeah.
Heavier atoms with more electrons
act
as stronger signals. They help us pinpoint their position and piece together the structure.
So it's like landmarks on a map,
Yeah.
guiding us towards the full picture.
I like that.
Yeah.
And all of this without computers.
Right.
Hodgkin was working with tools like Patterson synthesis, beavers, and lips and strips, literally strips of paper with calculations on them.
I can't even imagine.
So patience.
Yeah. It's mind-boggling. Like, what we can do in hours now would have taken them months.
It's incredible.
Of painstaking calculation.
Talk about dedication. And it led to some groundbreaking discoveries.
Yeah.
Cholesterol iodide. This was key in confirming the structure of sterols, which are essential components of cell membranes. So that means, for you, this is a better understanding of how your cells function at the most basic level.
Yeah.
Then there's the wartime challenge. Penicillin. Tiny amount to work with.
Oh, wow.
The pressure was on to determine its structure.
That must have been intense.
I know. Did they succeed?
They did. That was huge.
Yeah.
Paved the way for understanding how it works. Revolution in treating bacterial infections. I mean, think about the impact.
Countless lives.
Countless lives.
It's incredible how fundamental these discoveries were.
Yeah.
And she didn't stop there. Vitamin B12. Even more complex. And can you imagine the surprise when they finally uncover the structure?
Oh, yeah.
It was unlike anything they'd seen before. This intricate three-dimensional puzzle.
That was a massive leap forward and required collaboration with, people who had access to those early computers. It shows how technology plays a role in this kind of scientific progress.
It was a team effort for sure. Speaking of complex molecules, we can't forget about cephalosporin C. Another antibiotic with a fascinating structure.
Okay.
It actually has a four-membered ring.
Huh.
Quite unusual in nature.
Yeah.
And it originates from a similar source to penicillin.
Okay.
But in an oxidized form.
Yeah.
It really highlights how nature just builds on these similar blueprints to create diversity.
It's like evolution at the molecular level, right?
It is.
There's always something new to discover, even in seemingly familiar places.
Now let's dive into one of Hodgkin's most ingenious techniques.
Okay.
Anomalous dispersion.
Okay.
Used it to study cobiuric acid.
A precursor to vitamin B12.
Okay.
But before we get too deep, how would you explain anomalous dispersion?
Oh.
In simple terms.
Okay, so think about it this way. Certain atoms, like the cobalt in cobiuric acid, can change the phase of the X-rays that are passing through them.
And by carefully measuring these phase changes, we can pinpoint their location within the molecule.
So it's like those atoms are giving us extra clues.
Yeah. It's like they're leaving these unique fingerprints on the X-ray pattern.
That's pretty cool. Didn't they see this effect visually in the data?
They did. You can see it. Like this asymmetry in the X-ray reflections from the crystals.
Wow.
It's direct evidence of anomalous dispersion in action.
Like seeing the invisible world come into focus?
Yeah.
What's even more amazing, the electron density map they created.
Yeah.
So clear.
Really?
That they could see not just the chemical formula, but even the hydrogen atoms.
Wow. Talk about a high-resolution image. That's incredible.
Right.
Yeah.
And, just to double check,
Yeah.
they compared their structure to a synthetic nickel-corin derivative.
Oh, okay.
Made by Dunitz.
Okay.
Further confirming.
Multiple lines of evidence.
Exactly.
It's like having multiple witnesses corroborating the story.
That's right.
Okay, so they conquered coburic acid.
But what about B12 itself? The coenzyme plays a critical role in all these processes, but it was notoriously difficult to isolate, unstable, real challenge to study.
This is where X-ray crystallography really shines.
Yeah.
Allowed them to see something that nobody had anticipated. A direct link between the cobalt atom and the adenosyl residue.
Wow.
So this unexpected connection, it was a major aha moment.
So it wasn't just about the structure, it was about understanding how the pieces fit together.
That's right, that's right. This discovery really explained why it was so unstable, and that intricate geometry, it's key to understanding its function.
Yeah.
Particularly in transferring those methyl groups. It's like...
Form follows function.
Yeah.
It makes a lot of sense. But even with the structure, so many questions.
Oh, yeah.
Especially how it works within a living cell, right?
Right. We're talking about a process that's constantly in motion. X-ray crystallography, it gives us this snapshot. But to really understand the dance, we need to see them in action within the enzymes.
Right.
Yeah.
So it's like having the still photo versus the full-length movie.
Exactly.
Both valuable, but different parts of the story.
Totally.
And this brings us to Hodgkin's work on an even larger and more challenging molecule. Insulin. Key to regulating blood sugar.
Yeah.
We knew the chemical structure from Sanger's work.
Right.
But its exact mechanism, still a mystery. Even today, despite all this research.
True.
Still so much we don't understand.
That's the beauty of science, right?
Yeah.
Always more to explore.
Always more to explore. So Hodgkin discovered that it could actually crystallize in different forms.
Oh, wow.
And these forms are related to its function.
Interesting.
So, for example, it's dimeric, meaning two molecules bound together in acid salts.
Yeah.
But it potentially forms these larger aggregates in its cubic form.
Interesting.
And definitely when zinc is present. So the environment influences the shape.
Right.
And the shape influences how it works.
That's fascinating.
Isn't that interesting? And there's a historical connection here. Back in 1927.
Oh, wow.
Abel actually observed cubic insulin crystals.
That's amazing.
Decades before.
Yeah.
So even early observations can lay the groundwork for later discoveries.
It's true.
It's like a scientific echo across the years. But insulin presented new challenges.
Oh, yeah.
Introducing those heavy atoms.
Right.
Proved tricky.
Oh.
It was a real marathon.
Yeah.
Full of obstacles, exploring all sorts of techniques.
I can imagine.
Anomalous absorption effects.
Oh.
To try and crack the code.
Okay, so for those who don't know, anomalous absorption effect.
Or how would you explain that?
So basically, when X-rays hit certain atoms,
right,
they can be absorbed in a way that changes the intensity of the scattered beams. And, by analyzing those changes, you can glean more information about the positions of those atoms.
It's adding like another layer of detail
to
our map.
Yeah.
It's incredible how scientists find these subtle clues to piece together the puzzle.
It's amazing.
But I feel like we've been talking about Hodgkin for a while.
We have.
You ready to move on to Venki Ramakrishnan and the ribosome?
Let's do it.
Okay, buckle up. We're about to get molecular.
All right. Ribosome, it's like this molecular factory, It takes the instructions from your DNA and churns out proteins based on those blueprints.
It's a pretty important job.
Huge. It's a complex machine. 2 main subunits. And scientists have been working for years to try and unravel all its secrets.
It's a big one to tackle, for sure.
Yeah.
So Ramakrishnan, he started by focusing on the structures of individual ribosomal proteins.
Right.
Kind of like the building blocks of this machine.
Yeah.
But he realized that to really understand how it works, he needed to see the bigger picture.
Right.
He needed the structures of the entire subunits.
Like trying to figure out how a car works by looking at the engine, the wheels, seats separately. You got to see how it all comes together.
You need the whole system.
Yeah.
But crystallizing ribosomes?
Right.
Not easy.
No.
It's a large structure. Fragile. difficult to work with.
They're a real headache for a crystallographer.
How did he even manage to get those high-resolution structures?
This is where his ingenuity comes in. He made some real landmark achievements in obtaining, diffracting crystals, and his innovative use of a technique called multi-wavelength anomalous diffraction, or MADI for short.
MADI phasing.
Yeah.
Break it down for us non-crystallographers.
Okay, so imagine taking multiple X-ray pictures of the same crystal.
Okay.
But each picture is taken with a slightly different wavelength of light.
Okay.
So by analyzing those subtle differences in those images, you can pinpoint the locations of specific atoms.
Okay.
It's like using different colored lights to highlight different parts of a stage.
I see. It's about exploiting those subtle differences.
Yeah, exactly.
To extract more information.
Yeah.
Didn't he face some initial roadblocks with MADI phasing, though?
He did. And this is where the story gets really interesting.
Yeah.
Initially, he calculated that there wasn't enough methionine in the 30S subunit for MADI phasing to really work.
Oh, so what did he do? Just give up?
No, he had to think outside the box a little bit.
Yeah.
Find alternative atoms that would work.
Okay.
He ended up using lanthanides.
Interesting.
Which is a group of rare earth elements.
Okay.
And osmium hexamen. It was a real challenge. Classic example of scientific problem solving, adapting your approach when you face a challenge.
It's inspiring to see how a setback can lead to a more creative solution.
It can.
So besides atom phasing, are there other methods for determining the phases?
Yeah. There are a couple of other ones.
Okay.
Heavy atom clusters, molecular replacement.
Go ahead.
Heavy atom clusters. Think of it like attaching little tiny weights to specific parts of the molecule so they kind of stand out in that X-ray data.
Okay.
And then molecular replacement uses a previously solved structure as a starting point to help solve the new one.
So it's like having a reference guide.
Exactly.
To help you solve a puzzle.
Yeah.
So a whole toolkit of approaches to tackle these complex structures.
It's all about finding the right tool for the job.
Absolutely.
Yeah.
Let's dive into what they discovered about the 30S subunit.
Okay.
What did this structure reveal about the inner workings of the ribosome?
This was a major moment in ribosome research.
Okay.
30S subunit structure revealed a wealth of information.
Okay.
Some of it was really unexpected.
Like what?
Well, they could clearly see the right-handed double helical structure of the RNA within the subunit.
RNA, not DNA.
RNA. Crucial player.
Interesting.
Confirmed its important structural role.
Very cool.
And they were able to identify specific elements of the structure, helix 44, and figure out where those ribosomal proteins fit into the electron density.
So they're putting together this three-dimensional jigsaw puzzle.
Exactly.
Anything else surprising?
They did, and this was a major shift in our understanding of the ribosome. They discovered that the functional sites, where the real action happens, are primarily composed of RNA, not proteins. And the interface between the two subunits also dominated by RNA. This challenged the view at the time that proteins were the main players.
Wow. So RNA taking center stage?
RNA is the star.
Not just the supporting actor?
Not at all.
That's a big deal.
It was a game changer.
Yeah.
Of course, proteins still play a role.
Yeah.
But this highlighted RNA's significance.
Okay.
They also found that many of the proteins have these long tails that interact with the RNA.
Okay.
It's this intricate interplay.
So it's a delicate dance between RNA and proteins working together.
That's a good way to put it.
To build the molecules of life.
Yeah.
Now, the story of the 30S subunit doesn't end there, right?
No, it doesn't.
This structure helped us understand how antibiotics work.
Yes. 30S subunit opened up this new avenue for studying how antibiotics target bacteria.
So it's like having a blueprint of the battleground. It's showing us where those molecular weapons strike.
Yeah.
So they were able to see where the antibiotics bind.
Exactly. They analyzed something called difference Fourier maps, which highlight the changes in the electron density when that antibiotic is present.
So you're comparing before and after.
Yeah.
To see where it latches on.
You got it.
And one of their remarkable findings, the 30S subunit with 3 different antibiotics bound simultaneously.
3.
Streptomycin, spictenomycin, and paromomycin.
Wow, caught it in the middle of a molecular siege.
What did these structures tell us?
They provided a ton of information about how they interfere with bacterial protein synthesis.
Okay.
Streptomycin distorts the ribosome, leading to errors in decoding.
Okay.
Spectinomycin blocks the movement of tRNA, and then paramomycin, which we'll discuss a little later, gives us incredible insights into this process of decoding itself.
Decoding. You mean how the ribosome accurately translates the genetic code, making sure the right amino acids are linked together to build the proteins?
It's crucial for making sure the right protein is made, like making sure you follow the recipe correctly.
You mix up the ingredients, you get a different dish.
Exactly.
So what's the challenge with decoding? How does it ensure it's selecting the right tRNA?
So it's a remarkable process.
Yeah.
And it starts with the genetic code itself. Ever since we cracked the code, we've known that it's degenerate.
Degenerate.
Meaning multiple codons can code for the same amino acid.
Oh, so there's redundancy built in.
Exactly. And because there are fewer tRNAs than codons, Crick proposed the wobble hypothesis.
The wobble hypothesis.
Yes.
Doesn't it mean that some mismatches are tolerated at the third position of the codon?
You got it. But here's the thing. The energy difference between a correct base pair and an incorrect one isn't really enough to explain the accuracy. The ribosome gets it right almost every time.
So there must be something more to it.
There is.
It can't just blindly accept any tRNA that comes along.
No. Scientists began to suspect that the ribosome itself plays an active role.
Like a quality control inspector in that molecular factory.
Okay.
So early evidence for this came from those studies with streptomycin.
Okay.
Binds to the 30S subunit.
Yeah.
And increases errors in protein synthesis.
So by messing with the ribosome, it throws a wrench in the decoding process.
Exactly. And these findings led to this idea of a decoding center.
Okay.
Within the ribosome.
Right.
Where that pairing is carefully inspected.
So a proofreading mechanism.
That's the idea. But then the discovery of something called a Hirsch suppressor, this fidelity mutation.
Okay.
It's located far away from the tRNA in acodon. Challenged this view.
So a mutation far away affects the accuracy.
It does. And this led to an alternative view called kinetic proofreading.
Kinetic proofreading?
Yeah.
What's that?
In this model, the ribosome isn't directly inspecting the pairing.
Okay.
It's more about modulating the rates of various steps.
Okay.
It's more about controlling the timing of events.
Okay.
Than physically checking the base pairs. So like a series of hurdles.
Yeah.
And incorrect tRNA is more likely to trip over.
Exactly.
Right.
So incorrect tRNAs are more likely to dissociate before or after a key step, involving a protein factor called EF2 and the energy molecule GTP.
So controlling the flow of events.
That's it.
Okay. But if it's not directly inspecting, how can a mutation so far away affect accuracy?
It's a great question.
Yeah.
And it really highlights this interplay between structure and function.
Right.
Kinetic proofreading suggests that both the ribosome and the tRNA are somewhat passive.
Okay.
You know, players in this process.
Okay.
Mutations, even those far away, can influence the rate of GTP hydrolysis by EF2.
Okay.
That's a key step.
Yeah.
In this proofreading process.
Got it. So a delicate balance of speed and accuracy.
Exactly. But the story doesn't end there.
Okay.
As scientists dug deeper, they realized that that original view of the decoding center might not be entirely wrong.
So more to the story. It does actually inspect them directly.
It appears so. And the structure of the 30S subunit, along with those studies using paramomycin, provided some clues.
Paramomycin, the one that was bound to the ribosome along with those other two.
That's the one. The structure of the 30S subunit bound to paramomycin showed something really remarkable.
Okay.
The antibiotic stabilizes the conformation of two key bases, A1492 and A1493.
Okay.
In a way that suggests they can directly inspect that minor groove of the codon-anticodon helix.
So they're like molecular calipers checking for the right fit.
Exactly.
So it does have a way of double checking.
It does.
Not just relying on the timing.
Yeah.
Okay.
These two bases, along with another one called G530, seem to work together to sense the precise geometry of those base pairs.
Didn't you say it's super difficult to crystallize ribosomes with mRNA?
It is. But Ramakrishnan and his team made this really fortunate discovery.
Okay.
They realized that the A site of the 30S subunit, where decoding occurs, is exposed to these large solvent channels in the crystal.
So there's a way to sneak in the ingredients through a back door.
You got it.
Okay.
Allowed them to soak small RNA molecules that mimic mRNA and tRNA directly into the crystals.
So they set up this controlled environment to observe the interactions?
That's exactly right.
I love it.
Yeah.
What did they learn from these experiments?
Okay, so their findings were really groundbreaking. They showed that when a cognate tRNA...
Cognate meaning a perfect match?
Perfect match. binds to the 30S subunit.
Yeah.
It causes a change in the conformation of those 3 key bases.
Okay.
A1492, A1493, and G530.
Right.
So these bases, they fit into that minor groove of the codon-anticodon helix, but only if the base pairing is correct at those first 2 positions.
So they're checking for the right handshake.
Yeah.
Before allowing the tRNA to proceed.
Exactly.
I love it. This interaction provides a beautiful explanation for the wobble hypothesis.
It does.
Being strict about the first 2 positions.
Exactly.
More lenient at the wobble position.
That's right.
It's all about that balance between accuracy and flexibility.
Yeah.
But you also mentioned global conformational changes.
Right. So they discovered that cognate tRNA binding doesn't just cause those local changes in the decoding center.
Right.
It triggers this large-scale conformational change in the 30S subunit itself.
So it's not just about checking the code, it's about locking the correct tRNA into place.
Precisely.
Okay.
And this domain closure,
Yeah.
as we call it,
Okay.
only happens with a cognate tRNA unless paramomycin is present.
Paramomycin again.
It's a key player in this story.
I love it.
So this finding provided a really crucial link between structure and function.
Okay.
Kinetic studies had shown that cognate tRNAs accelerate the rate of GTP hydrolysis by EF2.
Right.
Paramomycin mimics this effect even with near-cognate tRNAs.
Near-cognate, what does that mean?
So a near-cognate tRNA is one that has a single mismatch
Okay.
with the codon.
So almost a perfect match.
Almost. And this is where paramomycin throws a wrench into things
Okay.
by stabilizing the ribosome. Yeah. In its closed conformation, even with that near-cognate tRNA, it allows the wrong amino acid to be added.
So it's like tricking the ribosome into accepting an imposter.
Exactly. And this highlights the importance of that domain closure for GTP hydrolysis.
Right, because that's the point of no return.
It is.
Once that happens.
The tRNA is
Okay.
committed. Yeah.
But why is domain closure so important for GTP hydrolysis?
To understand that, we need to zoom out.
Okay.
Look at the whole ribosome,
Yeah.
not just the 30S subunit.
Okay.
This is where cryo-EM comes into play.
Cryo-EM, the incredibly detailed 3D images.
You got it.
Yeah.
Cryo-EM studies had shown that the tRNA, when bound to the ribosome as part of this complex with EF2, is distorted. Bent out of shape.
Huh. So it's not just sitting there passively.
Not at all. So based on this observation, they proposed a model
Okay.
where the energy from that correct codon-anticodon pairing,
The handshake.
yeah, that perfect handshake,
Yeah.
helps to drive the domain closure. And this closure stabilizes the distorted tRNA.
Okay.
And this distortion promotes GTP hydrolysis by EF2.
So it's a chain reaction.
Yes.
tRNA binds,
Yeah.
ribosome closes,
Mm-hmm.
tRNA distorts
Yeah.
GTP hydrolysis.
That's it. Like a carefully choreographed molecular dance.
It's amazing.
Yeah.
And it explains why those mutations in the ribosome and tRNA can affect fidelity.
Exactly.
Because they can make it easier or harder to achieve that domain closure,
Right.
throwing off the timing.
Exactly.
Wow. These tiny molecular tweaks can have a huge impact.
They can.
How do they even get a structure of the whole ribosome with EF2 bound?
Huge challenge. Took years to overcome.
That's
Remember
it.
that protein L9?
The one they had to delete to study the 30S subunit?
That's the one. Well, they figured out how to get rid of it, or
Okay.
at least the part that was causing problems.
Okay.
They used some genetic techniques to just delete the gene.
So they did some molecular surgery.
Exactly.
Removed the troublesome bit.
Got it.
But kept the rest functional.
That's right. But here's another twist.
Okay.
Deleting that part of L9 actually prevented the ribosome from crystallizing.
Yeah.
Back to square one, had to start screening all over again.
Wow.
And this is where the story gets even more interesting.
Okay.
Years earlier, they had found some promising conditions for an EF2 complex, but they abandoned them.
They abandoned them. Why?
Well, the scientists working on that project left the lab.
Oh.
Research was put on hold.
Wow.
Really shows you how science is a collaborative effort, and sometimes progress depends on the right people being in the right place at the right time.
It's true.
Yeah.
So those crystallization conditions just disappeared into a lab notebook somewhere.
Thankfully, no. Years later, they stumbled upon a new crystal form of an EFG complex.
Okay.
Another GTPase factor, using conditions that were strikingly similar to those initial hits for the EF2 complex.
Oh, wow. What are the chances?
So they resurrected those old conditions,
Okay.
and they got crystals of the EF2 complex bound to the ribosome.
They did.
Yeah, but they had to use one more trick.
Okay.
They added a molecule called curamycin,
Curamycin.
which stalls the complex after GTP hydrolysis.
Okay.
It's like hitting the pause button,
Yeah.
allowing them to capture that snapshot.
So curamycin. Another antibiotic.
Yeah.
These molecules are really playing a starring role in unraveling these mysteries.
They are. And the structure they obtained with keramycin was really amazing, confirmed many of their hypotheses about the decoding pathway, and gave us a much deeper understanding of how it all works.
Okay, I'm on the edge of my seat. Tell me, what did this structure reveal? How does it all come together?
It really is a combination of years of research, confirms that sequence of events we've been discussing, recognizing the right codon, GTP hydrolysis, all of that. It all starts with the tRNA escorted to the ribosome by EF2,
Okay.
which is bound to GTP.
So EF2 is like a chaperone.
Yeah, making sure it gets to the right place.
Okay.
Once it arrives, the tRNA starts sampling different codons on the mRNA, kind of like trying on different shoes until it finds the perfect fit.
And if it's the right fit, that decoding center springs into action.
Yes.
Double checking everything's in order.
Those three bases, A1492, A1493, G530, they interact with the minor groove, but only if the base pairing is right at those first two positions.
The molecular handshake.
I like that. And that recognition triggers the 30S subunit to close up, bringing everything closer to EF2.
Okay.
Causes all these tiny little shifts in the structure, ultimately leading to a key interaction between the tRNA and EF2 being broken.
Like a chain reaction.
Yeah.
One change setting off another.
Exactly. And that's when the GTP molecule becomes exposed, making it vulnerable to hydrolysis. GTP hydrolysis, the point of no return.
Right.
Once that happens, EF2 changes shape, releasing the tRNA and the ribosome.
So the tRNA is finally free to move to the peptidyl transferase center.
Yes.
Add its amino acid.
That's
To
right.
the growing protein chain.
Exactly.
It's like it's passed all the tests.
Yeah.
And it's ready to contribute.
It's a beautiful system. And this structure with EF2 and keramycin allows us to really see these steps in such detail.
Amazing.
Yeah. Confirms what we thought we knew about decoding.
Power of X-ray crystallography.
It is. But remember, we're not just talking about structures.
Right. Function.
We're talking about function, the dynamic processes that make life possible.
Not just a static picture, right? It's about understanding how they move and interact, how they work together to carry out these essential tasks.
Exactly. Okay, I think we've explored Hodgkin's world
Yeah.
and Ramakrishnan's world.
Ready for a change of pace?
I think so. Let's talk about Woodward.
All right. Robert Burns Woodward.
A legend.
Chemical synthesis.
He wasn't just satisfied with observing molecules.
Okay.
He wanted to build them, create them from scratch.
So we're moving from observing to building.
We are. And we're going to focus on his work with cephalosporin C. Remember, we talked about it earlier.
Right. Antibiotic related to penicillin.
Yes. With that beta-lactam ring. Woodward took on this challenge that most people thought was impossible.
Really?
He decided to synthesize it from scratch, presented his unpublished work on it at his Nobel lecture in 1965.
What? Talk about confidence.
Yeah.
What made it so difficult? I mean, wasn't it similar enough to penicillin that they could have just used existing methods?
Well, it does share some features with penicillin, including that beta-lactam ring, which is really fragile.
Yeah.
But with cephalosporin C, you need to make sure all the atoms are arranged in a very specific way in three-dimensional space.
Oh, I see.
It's called stereospecific synthesis.
Okay.
And it's not easy.
It's like building a house of cards.
Yeah.
But each card has to be perfectly placed.
Exactly. To make things even more challenging.
Yeah.
He chose to start with L plus cysteine,
Okay.
an amino acid,
Right.
that already contains a chunk of that structure.
So you're starting the puzzle with a few key pieces in place.
Yeah.
But still tons of work to do.
Tons of work. But cysteine is a bit of a wild card. It's got all these reactive groups that could cause all sorts of problems.
Yeah.
Unwanted side reactions.
Right.
So they had to really carefully protect those groups. It's like putting molecular safety goggles on them.
I love it. So they could manipulate it without causing chaos.
That's right.
So they were able to control its reactivity.
They were. It was impressive.
Yeah.
They also had to introduce a nitrogen atom into a part of the molecule that's resistant to attack.
Right. Into that methylene group.
You're remembering the details. I try.
But they came up with this novel reaction using dimethyl azotocarboxylate.
Okay.
This allowed them to put that nitrogen atom exactly where they wanted it.
Wow.
In the correct orientation.
So they threaded the needle
They did.
with pinpoint accuracy.
While navigating a minefield of potential side reactions.
It's a testament to their mastery.
It is.
Okay, so they got the nitrogen in place.
Hmm.
What's next? Building the beta-lactam ring.
That's right. And that ring is so fragile.
Yeah. Prone to falling apart.
Right.
They had to handle it with molecular tweezers.
I can imagine.
carefully converting the molecules step by step.
Yeah.
Until they finally formed the beta-lactam ring.
I bet there was tension in the lab when they were working on that stuff.
Sure.
Did they confirm the structure?
Of course. They used X-ray crystallography
Of course.
to confirm the structure of the beta-lactam.
Smart.
Yeah. And then came the final stages of the synthesis. Adding more pieces, building out that core structure.
So building the foundation, then the walls, then the roof.
Exactly. And some clever tricks along the way. For example, using a protecting group that could be removed without damaging the beta-lactam ring.
Like a molecular parachute.
Yes.
So they could land safely at the destination.
Exactly. And after a series of carefully choreographed steps, they reached their goal.
Synthetic cephalosporin C.
They did.
They compared it with a natural compound.
They did perfect match. Even tested the antibacterial activity to be extra sure.
Wow. That is a remarkable story of scientific ingenuity.
It is.
Perseverance.
For sure.
Really makes you appreciate the complexity.
Yeah.
And the effort it takes to not just understand, but recreate them.
It shows you we can not only understand these intricate structures, but also become molecular architects ourselves.
That's a great way to put it. This has been quite the journey.
It has.
X-ray crystallography, chemical synthesis, seeing how they've revolutionized our understanding of molecules.
It's incredible.
From the smallest building blocks to the complex machinery of life.
The challenges, the breakthroughs.
The collaborative spirit.
It really is a testament to human curiosity.
And the pursuit of knowledge.
Absolutely.
It leaves us with a sense of wonder, doesn't it?
It really does.
So many secrets waiting to be unlocked. Who knows what we'll explore next?
What challenges we'll overcome?
I'm excited to find out.
Me too. And, you know, maybe you'll be the one to make the next big discovery.
You never know.
Keep diving deep. Keep asking those questions. Never lose that sense of wonder.
That's a great note to end on. Thanks for joining us on this deep dive.
Thanks for having me.
And we'll see you next time for another exploration of the
欢迎
fascinating
收听
world
我
of
们的深度解读。
大家好，
今天我们来聊聊3位诺贝尔奖得主多罗西·霍奇金、文吉·拉马克里什南，还有罗伯特·伍德沃德。我们想通过他们的研究，看看科学家是怎么，可以说是看见那些超级小又复杂的生命分子，又是怎么创造它们的。
对，这其实就是一次关于洞察分子结构和施展合成艺术的旅程。
了解生命的构造，然后还能亲手去合成。
是的，我们会从霍奇金怎么用 X 射线看结构开始，然后聊到核糖体这个巨大的生命工厂，最后再看看伍德沃德高超的合成技巧。
好，那咱们先说看见分子。多罗西·霍奇金那个年代可没有现在这么先进的显微镜吧？
没有，完全没有。
那他怎么去验证？比如说青霉素、维他命 B12这些复杂东西到底长什么样？化学家当时也就是推测。
他用的就是 X 射线晶体学。这个技术原理不复杂，就是用 X 射线穿过长得规整的分子晶体，
然后看衍射图案。
对，但是呢，你能测出来的是衍射点的亮度，就是强度，可有的关键信息丢了，就是相位。
相位，这个怎么理解？
打个比方，就像你听音乐，只知道声音有多大，但不知道它的音高是哆、是咪还是嗦
啊，那确实没法还原音乐本身了。
对，分子结构也是一样，没有相位就没法从衍射图反推出原子在哪儿，三维结构就出不来，这就是所谓的相位难题。
那霍奇金他是怎么搞定这个难题的？
他和他的前辈们想出了一些很巧妙的办法，比如在分子里藏个比较重的原子，像个路标一样。
重原子？
对，重原子，或者用一种叫 Patterson 函数的数学工具。这些方法就像给那个模糊的衍射图加上了坐标系，能一步一步把原子位置给拼出来。
听起来就好复杂啊！
是非常复杂，尤其是维他命 B12那个分子结构，极其扭曲，特别难。解析过程真的是一波三折，甚至还用上了当时刚刚起步的计算机帮忙。
所以他的工作等于是给了化学家一双能看见分子内部细节的眼睛。
没错，这不仅仅是技术上的突破，更是他惊人的洞察力和那种就是不放弃的坚持。这直接改变了我们对生命分子的认识，还有药物研发。
好，从单个分子，我们现在跳到一个更厉害的大家伙——
核糖体。
核糖体的工作让我们看到了生命是怎么按照遗传密码指令一步步造出蛋白质了。 这个机器得有多复杂啊？
复杂太多了。如果说维他命 B12 是个复杂的自行车零件，那核糖体，简直就是一艘航空母舰了。
哇，
解析难度是指数级上升的。你想想，让这么个庞然大物长出能用于 X 射线衍射的完美晶体，本身就难于登天。
然后相位难题也更严重了吗？
对，更复杂了。拉马克黎士南他们团队用了当时最前沿的技术，比如叫多波长反常衍射，简称 MAD。简单说，就像用好几种不同颜色的 X 射线去照射晶体，从不同角度收集信息，然后想办法把这些信息整合起来，解出那个相位。
那他们通过这个结构发现了什么特别关键的东西吗？
关键的发现非常多，最核心的是，他们精确地展示了核糖体是怎么读懂信使 mRNA 上面的遗传密码，然后挑出正确的转运 RNA、tRNA 来运送氨基酸的。
就是那个decode的过程。
对 decode。特别有意思的是，他们发现核糖体 RNA 上有2个特定的碱基，就是 A2192 和 A2193这2个，它们会像质检员一样伸出来。
质检员？
就是仔细检查 mRNA 上面的密码子和 tRNA 上面的反密码子是不是配对正确、严丝合缝，如果配错了，它就拒收。
哇，这么智能，保证了蛋白质合成的准确性，
极高的准确性。而且这些结构还清楚地显示了像链霉素、次环素这些抗生素，它们是怎么正好卡在核糖体工作的关键部位，让它瘫痪的。
这对抗生素研发肯定意义重大。
是的，为设计新的更有效的抗生素提供了非常具体的蓝图。
好，我们看完了结构，现在轮到创造了。罗伯特·伍德沃德他在偶基化学领域简直是神样的存在，
确实是大事。
他合成头孢归菌素 C 的成就被看作是合成的艺术品。他当时的思路说句特别有开创性，跟别人想的不一样。
对，伍德沃德的策略非常高明。头孢金素 C 和青霉素结构有点像，但都非常难合成。当时合成青霉素的路子效果不太好，伍德沃德就没跟着走。
那他怎么另辟蹊径的？
他的第一个妙招是从一个叫 L+ 半胱氨酸的小分子出发。这个选择太聪明了。
为什么呢？
因为这个半胱氨酸啊，它不仅提供了后面需要的大部分原子骨架，更关键的是，它天然就带有一个我们需要的特定手性中心。
手性就是分子的三维空间构型。
对，就是那个立体结构。这一下子就解决了合成中一个老大难的问题，怎么控制分子的精确三维形状，等于起点就占了优势。
但是半胱氨酸有个地方好像本来没啥反应活性，却需要在那儿引入一个氮原子，还要控制方向，这拼起来还是很难呢。
这就是挑战，也是伍德沃德厉害的地方。他没硬来，而是设计了一连串特别精巧的化学反应，像打组合拳一样。
组合拳？
就是保护不需要反应的地方，活化需要的反应地方，转换官能团，一步步迂回前进。最终呢，不仅成功的在那个惰性位置安上了一个氮原子，而且还保证了它的空间朝向完全正确。
这真是巧夺天工。
更绝的是，它成功构建了那个贝塔内镶胺环。这个环状结构是头孢菌素和青霉素的核心，但它本身非常不稳定，很容易坏掉。
对，听说这个环很脆弱。
能把它做出来，并且还能用它进行下一步反应，这真的体现了他对分子反应性那种炉火纯青的理解和控制能力。最后再把其他零件拼上去，拆掉保护集团，大功告成。
整个过程就像精心策划的一场战役。
没错，充满了战略性和创造性，真的是化学合成的艺术。
听下来，从霍基金开创性的看见分子，到拉马克黎施南揭示生命机器的精密运作，再到伍德沃德展示这种创造分子的艺术，我们确实看到了科学探索不断深入的这个壮丽途径。
是的，确实是。理解这些复杂分子的结构、功能，以及思考我们如何去创造它们，这不仅仅是满足了人类的好奇心，更重要的是，它直接推动了医学、生物技术的进步，实实在在地改善了我们的生活。那么，当我们已经能如此精细地理解和操控微观的分子世界了，您不妨可以想一想，未来还有哪些更加激动人心的可能性正等待着我们去探索、去实现呢？
这确实是个值得思考的问题。好的，非常感谢今天的分享，也感谢您的收听，期待下次与您继续深入探讨。
wirklich sehen. Also Nicht nur die Formel auf Papier, sondern ihre genaue 3D-Architektur. Atom für Atom.
Ja, genau das macht die Röntgenstrukturanalyse möglich. Eine faszinierende Technik.
Und heute schauen wir uns an, wie diese Methode unser Verständnis von Schlüsselmolekülen wie Penicillin, Vitamin B12 und sogar dem riesigen Ribosom komplett verändert hat.
Das sind wirklich Meilensteine.
Unsere Quellen erzählen da echte Detektivgeschichten auf molekularer Ebene. Wir wollen verstehen, wie man diese komplexen Strukturen geknackt hat und warum das so wichtig war.
Wobei der Clou ist, man sieht die Moleküle ja nicht direkt. Das ist wichtig zu verstehen.
Ah, okay. Wie dann?
Also, Kristalle von diesen Molekülen, die beugen Röntgenstrahlen. Das erzeugt dann so ein Muster aus Punkten, unterschiedlich hell.
Okay, ein Punktmuster.
Genau. Und aus der Helligkeit, da kennt man die Stärke der Wellen, die das Muster erzeugen, aber, und das ist der Knackpunkt, die Phaseninformation fehlt.
Phaseninformation?
Ja, also wie die Wellenberge und Täler zueinander verschoben sind. Ohne die Phasen gibt's kein Bild vom Molekül. Das ist das berühmte Phasenproblem.
Und das war lange die große Hürde, nehme ich an.
Absolut. Das zu lösen, das war die zentrale Herausforderung.
Okay, und wie hat man das dann geschafft? Gerade bei den ersten wirklich komplizierten Fällen?
Tja, da gab es einen ziemlich cleveren Trick. Man hat gezielt schwere Atome eingebaut.
Schwere Atome? Sowas wie Blei?
Eher Jod oder Kobalt zum Beispiel. Die streuen Röntgenstrahlen besonders stark und ihre Position im Kristall, die lässt sich oft leichter finden. Zum Beispiel mit bestimmten mathematischen Methoden, den Patterson-Synthesen. Diese schweren Atome waren dann sozusagen Leuchttürme.
Leuchttürme, okay.
Genau. An denen konnte man sich orientieren, um die Phasen der anderen, schwächeren Wellen zu bestimmen. Und so Schritt für Schritt wurde das ganze Molekül sichtbar.
Verstehe. Aber bei Penicillin, mitten im Krieg, da lief das nicht so glatt, oder? Ich habe gelesen, da gab es Probleme mit den Kristallen?
Richtig, genau. Die verschiedenen Penicillinsalze, die man hatte, die waren sich chemisch zwar sehr ähnlich, aber kristallographisch waren sie nicht isomorph.
Nicht isomorph?
Heißt? Ihre Kristallgitter waren nicht exakt baugleich genug. Da konnte man die einfache Schweratommethode nicht direkt anwenden.
Oh, ärgerlich.
Ja, sehr. Man musste mühsam unvollständige Bilder von verschiedenen Salzen kombinieren. Das war eine Heidenarbeit.
Und heute? Wäre das einfacher?
Oh ja. Heute könnte man das vorhandene Schwefelatom nutzen mit bestimmten Techniken oder gezielt ein Bromatom einbauen. Das ging dann später bei Cephalosporin C, einem Verwandten, auch viel besser.
Und dann kam Vitamin B12. Das ist ja nochmal eine ganz andere Hausnummer, oder? Viel komplexer. Und man wusste anfangs fast nichts über den Aufbau.
Ja, das war Dorothy Hodgkins Meisterstück. Wirklich. Eine riesige Struktur.
Hodgkin, genau.
Die Röntgenanalyse hat da sogar geholfen, die chemische Formel zu korrigieren. Das muss man sich mal vorstellen.
Wahnsinn. Und wie ging sie vor? Wieder mit schweren Atomen?
Wieder half das Kobaltatom als Startpunkt, als Anker. Dann hat man sich mit immer besseren Elektronendichtekarten langsam vorangetastet.
Langsam heißt?
Über Jahre. Ein Glücksfall war, dass man auch ein Abbauprodukt analysieren konnte. Das enthielt den zentralen Chorinring.
Chorinring? Nie gehört.
War damals auch völlig neu, ein hochkomplexes Ringsystem. Aber das wirklich Verblüffende, die Sensation, war die Entdeckung einer direkten Bindung zwischen dem Kobaltatom und einem Kohlenstoffatom im Coenzym.
Eine Metall-Kohlenstoffbindung in einem Naturstoff.
Genau das. Das war damals, unglaublich. Das hat gezeigt, diese Methode kann völlig Unerwartetes aufdecken. Dinge, die man sich chemisch kaum vorstellen konnte.
Was für eine Entwicklung. Von diesen Pionierarbeiten mit Rechenschiebern und mühsamer Rechnerei bis hin zu gigantischen Strukturen wie dem Ribosom, der Proteinfabrik der Zelle.
Absolut, da sind wir in einer ganz anderen Dimension.
Und hier wird es wirklich spannend, finde ich.
Ja, die Aufklärung der Ribosomstruktur, also durch Ramakrishnan und andere, die war nur möglich durch massive Fortschritte.
Welche Fortschritte zum Beispiel?
Man brauchte extrem helle Röntgenstrahlung aus Synchrotrons und viel bessere Phasenbestimmungstechniken. Oft hat man da schwere Atome wie Wolframcluster oder Lanthanide eingebaut.
Und was sieht man jetzt?
Man sieht jetzt im Detail, wie die Zelle die genetische Information abliest. Es gibt bestimmte Basen im Ribosom, man kennt sogar ihre Nummern, A1492, A1493 und G530. Die funktionieren wie Inspektoren.
Inspektoren.
Ja, die überprüfen an einer bestimmten Stelle, der A-Stelle, ob der genetische Code auf der Boten-RNA, das Codon, und das Liefermolekül für die Aminosäure, das tRNA-Antikodon, ob die wirklich exakt zusammenpassen. Millimeterarbeit auf atomarer Ebene.
Und dieses Detailwissen, das hat ja dann direkte Konsequenzen, zum Beispiel für Antibiotika, oder?
Ganz genau. Man sieht jetzt präzise, wo und wie bestimmte Antibiotika, sagen wir Streptomycin oder Tetrazyklin, an das Ribosom binden.
Und es damit lahmlegen.
Richtig, sie stören die Funktion dieser molekularen Maschine. Und dieses Wissen ist natürlich Gold wert, um zu verstehen, wie Resistenzen entstehen und vielleicht auch, wie man neue Medikamente entwickeln kann.
Es bestätigt sich also immer wieder, die Struktur ist der Schlüssel zur Funktion.
Immer wieder. Man muss die Architektur sehen, um die Funktion zu verstehen und um eingreifen zu können.
Was bedeutet das also für Sie da draußen? Wir können diese fundamentalen Bausteine des Lebens nicht nur sehen. Woodward hat ja später eindrucksvoll gezeigt, dass man Moleküle wie Cephalosporin C, wenn man die Struktur kennt, auch im Labor nachbauen kann, synthetisieren.
Genau. Wissen über Struktur ermöglicht Kontrolle, Synthese, Veränderung.
Und das führt uns, vielleicht als letzten Gedanken für heute, zu einer ziemlich faszinierenden Frage, die über unsere Quellen hinausgeht, oder? Wenn wir die Architektur des Lebens jetzt auf atomarer Ebene verstehen und sogar nachbauen oder verändern können, was hält uns eigentlich davon ab, eines Tages völlig neue molekulare Maschinen zu entwerfen?
Ja, gute Frage. Maschinen oder Funktionen, die vielleicht weit über das hinausgehen, was die Natur selbst dir hervorgebracht hat?
Genau. Eine spannende Vorstellung für
This time, you gave us quite the collection of sources. We've got scientific papers, journal articles, and even a historical account, all centered around Nobel Prize-winning discoveries.
I love it.
I know, right?
Yeah.
And get this, the real excitement for me is how this deep dive connects fields that at first glance seem totally different.
Totally.
I mean, we're talking particle physics, organic chemistry, and even, get this, the study of the human brain.
It's amazing how interconnected these seemingly separate fields can be.
Isn't it fascinating?
Yeah.
Like how discoveries in one area often pave the way for breakthroughs in another, sometimes even decades or even centuries later.
Yeah.
It's like a giant puzzle where each piece, no matter how small, contributes to the bigger picture.
Definitely.
Speaking of small,
let's
start our journey by zooming in on the tiniest particles in the universe. Quarks.
Oh, wow.
I know, right? We've got excerpts from particle physics papers here.
Okay.
NBL50.pdf and NBL79.pdf that talk about experiments smashing particles together at insane speeds.
Yeah, these experiments were truly revolutionary. Trying to figure out how a watch works by smashing it against a wall and studying the pieces. That's kind of what these physicists were doing.
Right.
But with protons and neutrons.
And NBL79.pdf mentions that the results actually caused quite a stir.
Oh, yeah.
Because they hinted at something unexpected.
Okay.
A point-like structure within the proton. Before this, scientists thought protons were more like fuzzy blobs.
Right.
So what was the significance of finding these smaller, more fundamental components?
It completely changed our understanding of matter's building blocks. Those point-like structures turned out to be quarks.
Right.
And their discovery, huge implications.
I can imagine.
It's not just about the tiny scale. It's about understanding what our universe is fundamentally made of and how it all began.
So how does knowing about quarks impact us in our everyday lives? It seems pretty abstract, pretty far removed from our daily experiences.
Think of it this way. The Big Bang, the formation of stars and galaxies, even the elements that make up you and me, all come down to those tiny quarks and the forces that govern their interactions.
So you're saying...
Understanding quarks is like understanding the very language of the universe.
Wow, that's a pretty amazing thought.
It is.
Okay, are you ready to jump from the vastness of the cosmos to the intricate world of molecules?
Absolutely, let's do it.
All right. Let's dive into the work of Jean-Pierre Sauvage, Sir J. Fraser Stoddart, and Bernard L. Feringa.
Right.
All Nobel laureates in chemistry.
Yes.
Our sources, NBL54.pdf, NBL58.pdf, and NBL64.pdf, highlight their incredible contributions to the field of molecular machines.
Molecular machines.
Now, when I hear molecular machines, I think of tiny robots
working
away at the nanoscale.
Yeah. You're closer than you think.
Really?
These chemists took inspiration from nature's own intricate machinery,
Oh,
like the proteins that power our cells,
right.
and they figured out how to design and build artificial molecular systems capable of controlled movement.
Wow. So they literally created
Again.
moving machines out of molecules.
They did.
That's incredible.
Yeah.
But before their work,
Yeah.
were chemists even thinking about this possibility?
You'd be surprised. NBL54.pdf mentions a distinct lack of interest from the chemistry community before Sauvage's groundbreaking work with mechanically interlocked molecules like catenanes and rotaxanes.
Cananes and rotaxanes.
And rotaxanes.
Those sound like something straight out of a science fiction novel.
They might sound complex.
Yeah.
But imagine linking rings together
Uh-huh.
or threading a bead onto a string.
Okay.
That's essentially what these molecules are like.
Okay.
But the real breakthrough, figuring out how to make them move in controlled ways using chemical or light-based triggers.
So we've gone from passively linked molecules
Right.
to ones that can move and potentially perform tasks.
Exactly.
That sounds like a pretty big leap.
It was a paradigm shift in chemistry. For the first time, scientists were able to create artificial molecular systems that mimicked the dynamic, functional machines found in nature.
Wow.
And this opened up a world of possibilities.
So what kind of possibilities are we talking about here?
Okay.
What are the real-world applications of these tiny molecular machines?
That's where things get really exciting. Imagine targeted drug delivery, where a molecular machine carries a drug directly to a specific cell in the body.
Oh, wow.
Or think about nanoscale sensors, capable of detecting minute changes in their environment.
Okay.
The potential applications are vast and still being explored.
It's incredible to think that we might one day have
I know, right?
tiny robots made of molecules working inside our bodies
Crazy.
to deliver medicine or monitor our health.
It's wild.
I know. But let's not get ahead of ourselves just yet.
Right.
Before we explore the potential applications of these molecular machines, let's shift our focus to an equally fascinating realm.
Let's do it.
The human brain.
The human brain.
It's time to dive into NBL 78.pdf, which focuses on the Nobel Prize-winning work of David H. Hubel and Torsten N. Wiesel.
Those guys.
These two scientists revolutionized our understanding of how our brains process visual information.
Fascinating stuff.
NBL 78.pdf mentions something called ocular dominance columns.
Okay.
What are those exactly, and what do they tell us about how we see?
Imagine opening up the visual cortex, the part of the brain that processes sight, and seeing stripes running across it, each stripe dedicated to processing information from either your left or right eye.
Really?
Those stripes are ocular dominance columns.
Oh, wow.
And their discovery, major breakthrough.
So there are actual physical structures in our brains dedicated to each eye.
Yeah.
That's wild. But how does this help us understand how we see?
Hubel and Wiesel's research revealed how our brains take input from both eyes and combine it to create a single coherent image of the world.
Okay.
They also discovered orientation selectivity, meaning different groups of neurons in the visual cortex respond to different orientations of lines and edges.
So it's like our brain has specialized teams of cells.
Exactly.
Each responsible for detecting a specific feature in the visual world.
That's a great way to put it.
That's incredibly intricate and organized.
It is.
But why is understanding this important for us?
These discoveries have far-reaching implications.
Like what?
Not only do they help us understand the basic mechanisms of vision, but they also pave the way for developing treatments for visual impairments.
That's amazing.
It is pretty amazing.
We've covered so much ground already.
We have.
From the building blocks of matter to molecular machines and the intricacies of the visual cortex. But I have a feeling this is just the beginning of our journey.
Oh, we're just getting started. I knew it. There's so much more to explore.
Absolutely.
Right.
And in the next part of our deep dive, we'll connect the dots between these seemingly disparate fields, revealing the hidden threads that weave them together.
I can't wait to see how these seemingly unrelated discoveries are actually part of a larger, interconnected web of knowledge.
It's going to be good.
Stay tuned as we continue our exploration.
All right. Welcome back. Last time we journeyed from the infinitesimally small world of quarks to the intricate mechanisms of molecular machines and the brain's visual cortex.
Yeah, we covered a lot of ground.
Now it's time to connect the dots. Are you ready to uncover the hidden links between these seemingly disparate fields?
Absolutely. I'm so eager to see how these seemingly isolated discoveries are actually intertwined.
All right, let's start with the tools of particle physics.
Okay.
Those massive particle accelerators, like the one described in NBL79.pdf, require incredible feats of engineering.
Right.
And rely on materials that can withstand extreme conditions. So particle physics relies on breakthroughs in material science to push the boundaries of exploration.
Exactly.
That makes sense. But where do molecular machines fit into all of this?
That's where things get really interesting. Could we use the principles of molecular machines with their ability to operate at the nanoscale to develop even more precise and sensitive detectors for particle physics experiments?
It's like using the tiniest tools imaginable to study the universe's biggest mysteries. I love that idea. But what about connections between these molecular machines and what we learned about the brain?
Now we're venturing into truly exciting territory. Imagine molecular machines designed to interact with neurons.
Okay.
Perhaps delivering drugs with incredible precision, or even helping to repair damaged brain tissue.
It's like something out of a science fiction novel. Tiny molecular machines operating inside the brain, helping to treat neurological disorders or enhance cognitive function.
It's pretty amazing stuff, and the flow of inspiration can go both ways.
Oh, so.
Understanding how the brain processes information could lead to new algorithms for analyzing the massive amounts of data generated by particle physics experiments.
So it's a two-way street. Each field can learn from and inspire the other in unexpected ways.
Exactly.
Is there anything in our source material that specifically highlights this interconnectedness?
There is. In NBL78.pdf, Nobel and Wiesel, after their discovery of ocular dominance columns, write, "An entire system has opened up whose existence we were previously quite unaware of." This perfectly captures how each breakthrough can open doors to entirely new avenues of exploration,
Right.
often in fields we might never have imagined.
It's a beautiful reminder that knowledge is a constantly evolving landscape, and that every discovery can lead to unexpected connections and new mysteries to unravel.
Absolutely. And it emphasizes the importance of interdisciplinary thinking.
I see.
We need scientists who can see the connections between fields, who can bring together ideas from different domains to solve complex problems.
It's like assembling a team of superheroes, each with their unique superpowers, to tackle a challenge that no one could handle alone.
Exactly.
But let's not forget that even superheroes have to grapple with ethical dilemmas.
You're right.
What about the challenges and ethical considerations that come with these scientific advancements?
Great point. We can't just focus on the exciting possibilities without considering the potential risks and challenges.
Right.
Let's turn to NBL51.pdf, which delves into a rather unsettling topic: unconventional viruses and their role in diseases like Kuru.
Okay, this sounds a bit intense.
It is.
Can you give us some background on Kuru and why these unconventional viruses are so concerning?
Kuru was a devastating prion disease that affected the 4 people of Papua New Guinea.
Okay.
It was spread through ritualistic cannibalism.
Oh, wow.
Specifically, the consumption of deceased relatives' brains. What makes prion diseases so unique and challenging is that they're caused by misfolded proteins, not bacteria or viruses.
So it's like a domino effect, but at the molecular level. One misfolded protein triggers others to misfold, leading to a chain reaction that damages brain tissue.
Precisely. And what makes prions even more challenging is their resilience. They're resistant to many conventional sterilization methods, making them incredibly difficult to eliminate.
Oh, wow.
NBL51.pdf even mentions that these unconventional viruses are characterized by an absence of an immune response.
Really?
Our bodies don't seem to recognize them as threats.
So not only are they incredibly resilient, but they also manage to evade our body's natural defenses. That's a frightening combination.
It is.
Does the source offer any insight into potential treatments or prevention strategies?
Unfortunately, there's still no cure for prion diseases.
Oh, wow.
The source highlights the critical need for further research to understand the mechanisms of prion propagation and to develop effective therapies.
I see.
It's a sobering reminder that despite the incredible progress we've made, there are still formidable challenges in the world of science and medicine.
It underscores the importance of continued scientific investigation, even in the face of daunting obstacles.
Definitely.
But with powerful new technologies like molecular machines, it seems like ethical considerations become even more critical.
Oh, absolutely.
Are there any specific concerns highlighted in our sources?
NBL64.pdf touches on the concept of photopharmacology, using light to activate or deactivate drugs.
Interesting.
This holds tremendous potential for targeted therapies.
Right.
But it also raises questions about safety, unintended consequences, and the potential for misuse.
You're right. We need to be cautious and thoughtful in our approach.
Absolutely. And now let's shift gears and explore the work of Arturi Ilmari Virtanen, a Finnish chemist who revolutionized agriculture.
Okay, so we're going from the complexities of the brain and prion diseases to the world of agriculture. I'm curious to see how this all ties together. What exactly did Virtanen discover?
Virtanen was captivated by the process of nitrogen fixation, the conversion of atmospheric nitrogen into a form that plants can use. He focused on the symbiotic relationship between legumes and certain bacteria that live in their root nodules, acting as tiny nitrogen factories.
So these bacteria essentially help plants access the nitrogen they need to grow. That's incredible. But how did Virtanen harness this natural process to benefit agriculture?
He developed a method for preserving fodder.
Fodder.
Which is essentially dried plant material used to feed livestock.
Okay.
He discovered that by carefully controlling the pH of the fodder during preservation, he could prevent the breakdown of proteins and preserve the nitrogen content.
So he found a way to make the fodder more nutritious for livestock.
Exactly.
That's brilliant. But what's the larger impact of Virtanen's work? Why should our listener care about fodder preservation?
His discoveries had a huge impact on agriculture, especially in countries with limited arable land.
Oh, right.
His methods increased crop yields, improved the quality of livestock feed, and ultimately helped to address food security concerns around the world.
It's amazing how a seemingly simple discovery about preserving fodder can ripple outward to affect the lives of millions.
Isn't it?
It's a reminder that scientific breakthroughs can come from unexpected places and have far-reaching consequences.
Absolutely. And that's what makes this exploration of scientific discovery so captivating. It's a journey full of unexpected twists and turns, and every step forward can lead to groundbreaking innovations that shape our world.
We've certainly uncovered some fascinating connections today, haven't we?
We have.
From the building blocks of matter to the complexities of the brain and even the ingenious strategies of bacteria.
So many connections.
It's clear that knowledge is interconnected in surprising and often profound ways.
It is.
But our journey isn't over yet. There are still more mind-bending discoveries to explore, right?
Absolutely. Get ready for the final leg of our deep dive as we explore the frontiers of quantum physics, laser technology, and some truly mind-boggling concepts that will push the boundaries of what we know about the universe and ourselves.
We're back for the final part of our deep dive. So far, we've explored the interconnectedness of scientific knowledge, from the heart of matter to the mysteries of the mind, and even delved into the world of unconventional viruses and agricultural innovations.
It's been quite a journey.
Now we're diving into some seriously mind-bending concepts: quantum physics and laser technology.
These fields are all about pushing the boundaries of what we know about the universe and ourselves.
And the sources you provided, NBL53.pdf and NBL59.pdf, highlight the incredible work of physicists who achieved mind-blowing levels of precision in their measurements.
Absolutely.
Leading to incredible advances in our understanding of light and matter.
It's amazing what they accomplished.
I'm intrigued. NBL59.pdf mentioned something called a maser. Is that related to lasers?
That's a great observation. You're right. They are related.
Okay.
A maser is like a laser.
Yeah.
But it operates at microwave frequencies instead of visible light.
Okay.
Both produce highly focused beams of electromagnetic radiation. But they have different applications.
So what makes lasers and masers such valuable tools for scientists? What can they do that other tools can't?
It all comes down to coherence.
Coherence?
The waves in a laser or maser beam are all in sync, like a perfectly choreographed dance.
Oh, wow.
This allows for incredibly precise measurements and manipulation of matter at the atomic and molecular level.
It's like having a ruler that can measure things smaller than an atom.
Yeah.
That's amazing. But how do these concepts connect back to what we've already discussed?
Right.
Could we use lasers and masers to interact with those molecular machines we talked about earlier?
That's a brilliant question, and the answer is yes. Imagine using lasers or masers to control the movements of molecular machines with even greater precision.
Oh,
It would be like taking those tiny machines from manual to automatic transmission.
right.
And it's not just science fiction. NBL53.pdf highlights the use of lasers to cool atoms to incredibly low temperatures, a technique that has revolutionized atomic physics.
Why would scientists want to freeze atoms? It seems counterintuitive. Wouldn't that make them harder to study?
It might seem that way,
Yeah.
but at these ultra-low temperatures, atoms behave in ways that defy our everyday understanding of physics.
Oh, wow.
They start to exhibit quantum properties that are usually hidden from view. It's like opening a window into a hidden world where the rules of classical physics no longer apply.
This is mind-blowing. From smashing particles to freezing atoms and building molecular machines, this deep dive has taken us on an incredible journey through the frontiers of scientific discovery.
And through it all, we've seen how knowledge is interconnected.
We have.
Breakthroughs in one field can inspire advancements in others, often in the most unexpected ways.
Absolutely.
It's a testament to human curiosity, ingenuity, and the relentless pursuit of understanding the universe and our place within it.
It's truly inspiring to see how these seemingly unrelated discoveries are actually part of a larger, interconnected tapestry of knowledge.
It really is.
As we wrap up this deep dive, I'm left with a sense of wonder and excitement about what the future holds.
Me too.
What new connections and breakthroughs are waiting to be discovered?
Who knows?
What mysteries will we unravel?
It's exciting to think about.
And to our listener, we encourage you to keep exploring.
Absolutely.
Keep questioning.
Yes.
And keep diving deep into the world of knowledge. You never know what amazing connections and insights you might uncover.
You never know.
Until
我
next
们
time.
是怎么看见那些肉眼根本看不见的世界的？比如说构成万物的原子，或者我们鼻子怎么闻到味道，这些东西其实都藏在咱们视线之外。
对，很多东西我们是感知到，但不直接看到。
是的，今天呢，我们拿到了一些很有意思的资料，讲的就是科学家怎么一步步揭开这些微观世界的秘密。咱们的任务就是一起钻研一下这些资料，看看这些发现到底是怎么回事儿。准备好了吗？
准备好了。这确实是一趟挺迷人的旅程，跨越了不同的科学领域。核心就是理解那些让我们能看见看不见的关键技术，还有那些思维上的突破。
好，那咱们就先从看得更小、更清楚这儿说起吧。我们都知道，普通的光学显微镜它有极限，
对，物理极限，波长决定的。
但科学家肯定不满足于这个，
是吧？然后就有了电子显微镜。我看资料里说，最早 Nobel 和 Ruska 他们尝试用电子束成像。
是的，他们证明了用电子束和磁场来成像是可行的。虽然一开始有个大难题，
什么难题？
就是那个标本损伤。电子束能量高，容易把相品打坏，特别是生物相品。但这确实指明了一个全新的方向。
我记得资料里还提到一个挺有意思的转折，就是当他们意识到电子也有波长，会不会也限制分辨率的时候，好像有点小失望。
对，一开始有过那么一点点疑虑，觉得哎呀，是不是白费劲了？
哈哈。
但很快他们一算，发现电子的波长其实比可见光短太多了，
短很多很多。
对，极短。就意味着理论上电子显微镜的分辨率潜力是惊人的。
哦，原来是这样。
这个认识一下子就极大的推动了技术发展，最后真的让我们能直接看到单个的分子了。
就像资料里说的那个 Zemlin 他们拍到的石蜡晶体图像。
没错，非常清晰。这不只是看得更小了，是真的打开了分子层面的观察窗口。
不过对于那些特别脆弱的，比如说细胞里的核糖体这种生物大分子，直接用电子束观察是不是还是很难？
对，还是那个损伤问题。所以后来就发展出了冷冻电镜技术，就是 cryo-EM。
冷冻电镜就是把东西冻起来看。
差不多这个意思。像 Hope 和 Jonas 他们就是这方面的先驱。简单说，就是把相品用极快的速度冷冻在一种玻璃态的冰里面。
玻璃态的冰？
对，非晶体的冰，这样能最大程度保持相品结构，然后在极低的温度下观察。
低温？这样就能减少辐射伤了吗？
正是，低温能大大减缓损伤过程，而且这还能把分子的运动给定格住，看到它们某个瞬间的构想。
哇，定格，这简直是革命性的呀！
的确是。让我们能以前所未有的清晰度看到这些生命机器是怎么工作的。
太厉害了！好，那我们从分子世界再往里走到更小的地方，原子核。
原子核那更小了。
是啊，那个我们又是怎么探到它内部的呢？
这个最早是卢瑟福，他用那个 阿尔法粒子散射实验
啊，我知道那个精婆试验。
对，就像是在些屋子里扔小球，听撞到什么东西怎么弹回来，来判断里面大概有什么，多大。它就是用这个大致估算了原子核的大小。
只是大致估算，那要更精确呢？
要更精确的图像，就需要更强大的探针，这就是高能电子了。
这就是所谓的电子散射技术吧？用高能电子当子弹去打原子核。
没错，就是这个原理。通过非常精确的测量这些电子是怎么被原子核散射开的，就能反推出原子核内部电荷是怎么分布的。
哦，能画出分布图来。
可以这么说。这些实验还带来一些挺有意思的发现，比如大多数比较重的原子核，它中心的密度好像都差不多。
都差不多。
对，近似恒定。
但反倒是像氢、氦这种最轻的原子核，中心的密度特别高。
这有点反直觉啊。
是吧？
这背后就暗示了原子核内部作用力，也就是核力的复杂性。那原子核里面的质子和中子呢？它们本身是最小单位了吗？还是说它们里面还有东西？
这就问到更深层次的问题了。一开始大家都以为质子、中子就是基本粒子了，不能再分了，
对吧？
后来一种叫深度非反性散射的实验，彻底改变了看法。
深度非反性散射听起来就很厉害。
哈哈哈，你可以想象一下，不是用电子轻轻的去探测，而是用能量极高极高的电子去猛烈的撞击质子和中子。
猛烈撞击，像是用个小锤子去砸。
对，有点那个意思，像用一个分辨率极高的显微锤子去敲。
结果呢？敲出匣来了。
结果非常非常惊人。科学家发现，电子散射的方式就好像，它们撞到了质子内部一些更小的硬的点状物上。
点状物不是撞在一个均匀的东西上？
对，不是一个均匀的整体。就像你打碎一个桃子，发现里面有个更硬的核儿。
我明白了。
这种现象，这种特定的散射模式，就是我们现在说的夸克存在的非常强有力的证据。
夸克，原来是这么来的。
是的，这证明了质子和中子，它们也不是基本粒子，内部还有结构。这绝对是粒子物理学的一个巨大飞跃。
太不可思议了，真的是一层一层往下挖。好，我们从这个极小极小的粒子世界再跳出来，看看生物感官领域。
换个尺度。
对，比如说我们闻气味儿，这个过程看起来好像挺简单的，但背后是不是也藏着什么精密的编码系统？
绝对是，而且非常精妙。你想想，我们能分辨出成千上万种不同的气味。
对呀，花香、果香、咖啡香。
这个奥秘就在我们鼻子里的嗅觉受体，简称 ORS。关键的发现，比如 Melnick 他们做的研究揭示了什么呢？就是识别某一种气味，其实不是靠单一类型的受体，
不是一个受体对一个气味，
不是那么简单，而是需要一组特定的嗅觉受体组合被同时激活。
一组组合？
对，就像弹钢琴的和弦儿一样，不是单个音符，是几个音符组合起来才代表一个特定的和弦儿，一种特定的感觉。
哇，也就是说，每一种气味分子就像一个密码，需要一组特定的受体钥匙组合才能打开。
可以这么理解，非常形象。但更有意思的是，大脑是怎么处理这些信号的？
对，信号传到大脑之后呢？
表达同一种嗅觉受体的那些神经元，不管它们分散在鼻腔嗅上皮的哪个位置，这挺神奇的。它们的神经末梢，就是那个轴突，最后都会汇聚到大脑嗅球里。一两个非常特定的位置相对固定的区域。
等一下，就是说，所有表达同一款钥匙的神经元，最后都跑到大脑里同一地方去了。
没错，这些特定的汇聚区域就叫做嗅小球。
嗅小球？
对，所以这就等于是为每一种受体在大脑的嗅球层面建立了一个专属的着陆点。
天哪，这难以置信呢！就像给每种受体分配了一个固定的门牌号。
确实是这样。这就形成了一张极其精确的气味地图，不同的气味激活不同的嗅小球组合，大脑就通过解读这张地图上的激活模式来识别你闻到的是什么。
哇，
这种组织方式的精确性和那种内在的优雅，实在是让人惊叹。
真是太精妙了。下次闻到咖啡香的时候，我得想想大脑里这张地图是怎么亮的。
哈哈，对，可以想象一下那个复杂的神经活动。
好，我们这次的探索真的是，跨越了巨大的尺度啊。
是啊， 从分子到原子核，然后再到大脑。
对，从电子显微镜下的分子结构，到粒子加速器揭示的亚原子世界，再到大脑里面这个精巧的感觉编码，每一次深入都让人觉得哇，科学探索的力量真是太惊人了。
的确如此，这些资料看下来，其实共同揭示了一个主题，就是人类怎么凭借自己的智慧和创造力，不断地发明新的眼睛。
新的眼睛。
对，去突破我们自身感官的局限，去理解那些隐藏在表面之下更深层次的现实，它到底是怎么运作的。
那么最后留给大家的一个思考题就是，放眼未来，我们还能设计出什么样的更强大的探测器，或者说更厉害的显微镜？
未来会怎样？
对，用它们去揭开哪些我们今天可能甚至还完全无法想象的隐藏在现实之下的未知层面。
Thanks for listening. We're glad you were able to tune into this podcast. Once again, this is The New Chemist, where we discuss chemistry, which simply put is the science of change, as well as the other sciences, careers, community, research, and COVID-19. Thanks again for listening. Note, the views on this podcast represent those of my guests and I.